first, of the nurses who hold the baby, or of the mother also, but it's now believed that those first seconds might actually be important in our life. We can tell babies 10 minutes after they were born, we can tell them apart by looking at their skin bacteria. They're different between the cesarean born and the vaginally born bacteria. And those first seconds might have an influence later on in life. Although some cesarean sections, of course, are medically indicated, and so there might be something wrong with the mother or with the pregnancy that induced these cesarean sections. So it's very hard to tease apart those effects from the actual birth delivery mode in determining, in their effect of determining our microbiome. But it is believed that those first seconds might actually be important in the rest of our life and might have an association with things like allergy or obesity even. But it's very hard to do these type of statistical analyses in humans because everybody will have a different life history, and we cannot really do experiments like we could do in animals, where you can have all the animals experience the same food and the same life circumstances. But after those, so once we're about 3 years old, we'll have an adult-like microbiome. And that microbiome is actually relatively stable in the rest of our life. This is an experiment that was done in 2 male adults, about 30 years old, scientists, who sampled their stool samples for daily for about a year. You can see person A on the left and person B on the right. And you can see their gut microbiome followed over time, shown by these different colors. So every color is a different bacterial group, and you can see the relative abundances of each bacterial group over time. And there, you can first see that person A has a different microbiome than person B. They're both about 30 years old, both omnivorous, both active young men who, are still very different in terms of their gut microbiome. But you can also see that although their gut microbiome was relatively stable for about a year, there's a couple of changes. You can see that better on the plot below that shows the change with respect to the starting point. And you can see that person A had the first third of that year, that had a big life event. They actually moved to I think a country in Asia, lived there for 1 or 2 months, and then moved back. And although they weren't sick, you can see his microbiome changed a lot. When he moved back, it changed back to the old situation. Person B had another event in that year. He had a case of food poisoning with salmonella. And you can see that best on the plot, the lower plot, on the last third of his year. You can see that big change. So he had some food poisoning and he recovered. He didn't have antibiotics, he recovered quickly. But you can see that after that event, his microbiome actually changed. It did not go back to the old situation, although he didn't really notice that he felt any different. And so there's probably different states in which our gut microbiome can live. There's many different states that are healthy, like, you can get coffee at a Phil's and a Starbucks. Like it's still a coffee shop, but they look very different and they have some different brands. So similarly, you can probably have different communities in your gut that are all associated with health. They're just different from each other. And although they're relatively stable for periods of months, they can change with important life events like the food poisoning or moving to a different country. And there was a recent study that was done about a couple of months ago and was published in Science. There were 2 papers in Science that looked at which factor are most associated with the composition of our microbiome. And surprisingly, it was the Bristol stool chart, which is a very wonderful thing that you have to look up. It's basically describe how your stool looks like. And I have a coffee mug with a Bristol stool chart, so it has all the different phases of poop, like a long snake or different blobs, or you can imagine. And so describing the poop of the day that it was sampled was most associated with the composition. So basically how fluid it is or how solid. But other important factors that were associated with determining the composition are our diet, which is not surprising. So what we eat probably has a huge influence on our microbiome. But it's also believed that things like our genetics might play a role too. But diet is probably one of the largest influences. So certain microbes were associated, for example, with eating yogurt or with eating chocolate. So those there were a couple of really interesting associations that came up, where one particular bug was just more present in people who like dark chocolate. So it's very surprising insights that we didn't have a year ago. I think that's a really exciting topic that there's still a lot to learn about. How diet, what you ate today, probably will affect the bugs that live in your gut. So looking at that, how do the microbiomes of people who live in very different circumstances than most of us compare to Western gut microbiome? So there have been too few studies of that, but that's probably where we can learn the most. So I particularly like this study from the Hadza, which is a group in Tanzania, who live probably very similar to the way all humans lived about 10,000 years ago. They don't grow crops, they don't have farm animals, but they hunt and they gather. Basically, the men hunt. They hunt for raccoons and goats and wild pigs and deer. And the women will collect tubers and honey and berries. But their main diet consists of a lot of fiber stuff like tubers and leaves. So they eat very different than we do. They don't eat any sugars or very carbohydrates from crops like grains or anything like that. And they'll eat occasionally meat when the man has had some success in their hunt. They'll occasionally eat some meat. But it's a very different diet, and they also live in very nomadic type of circumstances. So they have much more contact with dirt and ground than we have. And their gut microbiome indeed reflects that. They have a very different microbiome. So in this graph on the left part, you see the Hadza, and they were compared to a group of Italians. And you can see that they have very different gut microbial groups. Although at the bottom, maybe the colors are very similar. Let me bring up the next one. The top part of the graphs are very different. You can definitely tell them apart. They have for example, the Italians have a lot of bifidobacterium in the blue, which are believed to be a sign of health. However, the Hadza, who are also healthy, don't have that bacterium at all. And so they have spirochetes, which we Westerners never have in our gut microbiome. And so they're healthy people, they are, but they have very different microbiomes. And those studies shattered insights we thought we knew what a healthy microbiome was defined by, but we now know that's not the case. You can have a very different microbiome if you live like the Hadza, and they still are healthy people. And so if you compare worldwide gut communities, you can see that that the Western or urban lifestyle is associated with a very different microbial composition than people who live a more agricultural or hunter-gatherer lifestyle. So if you think about this, our Western lifestyle probably doesn't bring us into contact with a lot of microbes. We mainly live indoors. We don't, most of us don't dig in the dirt or eat unwashed food. We live in urban circumstances with not a lot of access to plants or dirt. We regularly take antibiotics. Some of us use a lot of hygiene products, trying to sterilize our hands, for example. It's not going to happen. It's still going to be there. But it does have an effect. And some people even do colon cleanses, where they try to clean out their colon, basically wipe out their gut community. And what happens after that? Maybe they'll lose some bacteria. Maybe they'll never come back. And you might end up with a less diverse microbiome. It's believed that gut diversity is a good sign of health, and so if you get rid of all these bacteria by taking antibiotics or colon cleaning, you might lose some of that beautiful diversity that you're supposed to have. And that even starts at exposure to less bacteria already starts during birth, where some people are born by cesarean section. Some babies are fed with formula that doesn't contain the bacteria that breast milk fed babies will get. And again, antibiotics. By the age of 10, most American kids will have received 10 courses of antibiotics in their life. So every year they get a lot of antibiotics, maybe for an ear infection or something else. And of course you want to treat the baby, but it has collateral damage. It will also damage the bacteria in their guts, and that might have an effect later on in life. And Marty Blazer in the middle has a theory that those courses of antibiotics that most of the children get here in the US might be associated with obesity, with increased occurrence of diseases such as maybe autism or allergy. And these are hypotheses, they're not proven, but there might be a link there because our gut microbes are affected by every antibiotic course we take. Of course, having said that, there are cases in which you want to take antibiotics. I've had a big skin infection, and I thought, okay, I'm not going to take antibiotics, I'm not going to take anything for my gut to harm my gut microbes. But I got really sick and I got feverish. I'm like, okay, I should probably take something because I don't want to die. So there are definitely cases where you should take antibiotics, but maybe it's good to think about it and think if you really need it. I'm going to skip over a couple of the slides. These are some effects of the antibiotics, and these are some effects on not eating a lot of fiber. In both cases, taking antibiotics or not eating fiber have been found in animal experiments and in humans to be associated with less diversity. And you can also see it on the middle again, where some people from agricultural communities are compared to the bacterial diversity in people with an urban lifestyle. And the urban lifestyle, the Western lifestyle, is associated with less diverse, so that means less species in our gut. And you can compare that to, on the left, an urban lifestyle, or if you think about a park that only has a couple of different plants, like maybe has an oak tree or 10 oak trees and some grass, but there's not a lot of different plant types in there. And then if you think about a jungle that has like thousands of different species of plants. So similarly, our gut communities, if you have a Western lifestyle, have less diversity than people who are more in contact with dirt and eat more fiber. So to increase your microbial diversity, the main thing we think is important is eating fiber. And you can eat that by eating more vegetables, less meat and less carbs, and especially some very fibrous and raw vegetables. And if you even don't like vegetables, think about feeding your microbes. How about yourself? And I like cake much more than vegetables. But if you think about feeding your microbes, I don't know, it makes me feel better about eating a salad. And think about using less antibiotics if possible. Gardening apparently is very good because it brings us in contact with soil. I'm not preaching that we should all eat mouthfuls of dirt, but it's probably okay to occasionally have some dirt in your hands, or maybe not wash your carrots, to the extreme. Get a little bit of dirt, that's probably fine if you're healthy and not immunocompromised, I should add. So keeping pets is associated with a richer diversity. You can actually tell people who have pets, who can tell they have a slightly different microbial community in their gut. So having a pet affects the bacteria we have. So it's okay if the dog licks in your face, it's fine. Outdoor activities and eating fermented foods. So a very popular thing at Human Microbiome, where I work, is drinking kombucha, which is a fermented tea that is covered in sort of a layer with yeast and bacteria, but also other fermented foods like yogurts and kefir and sauerkraut. Those things are probably very good for us. They'll add to the diversity of the microbes we have in our gut. So, sort of a summary, if you think about the gut as partly small intestine where we absorb our food, humans absorb most of the food we can digest, then the large intestine is a vessel where the bacteria live and where they can digest what we cannot digest. So if we eat that cake that I like, it's a beautiful cake, that part gets absorbed in the small intestine. That's the easy accessible sugars. While the things we might not enjoy that much, the fibers, are passing through the first part of the small intestine, end up in the large intestine. That is the part where they can be digested by the bacteria. And that, then the bacteria they break down these fibers, break them down into small chain fatty acids, and those molecules actually are also absorbed by the human body, and those are the molecules that are believed to have effects on depression, on our functioning of our whole body, and also on feelings of satiety, so feeling that we have eaten enough. That leads to the gut-brain connection, which is sort of a very popular theme now in human microbiome research, where it's believed that we know that the gut is innervated by a lot of nerves. So there's a lot of connections between the brain and the gut in terms of nerve connections. But there's probably also a connection the other way around, where the gut has an effect on the brain. This is a very new and exciting area, so there's not yet much known, but there's definitely some groups who are trying to get some results in animals. Again, animals that are grown up without bacteria have a particular structure in the brain that is not as well developed as mice that were growing up with bacteria. There are also, those germ-free mice are also, they have different behavioral problems or different behavioral capacities than mice that grew up with bacteria. Some of them, it depends a little bit on the mouse strain, some of them do a lot of repetitive grooming, and a lot of these symptoms are similar to what we see in autism, like repetitive behavior, less communicative with each other. So a lot of the patterns we see in mice that grow up without bacteria are similar to autism. So there might be a lead that maybe the way that we grow up now is too sterile. We're not growing up with a lot of bacteria, and maybe that might be the cause for autism. Of course, a lot of these questions are very hard to answer in humans, and I'm not saying that that might be a true finding, but it's something to, it's one of the things that tells us that the gut microbiome is probably connected to a lot of different health conditions that we now know in humans, and that we still don't understand how they happen. We still don't really know if it's a particular bug or like a very vague number of bacterial cells that are associated with these diseases, but there are more and more studies that point towards the important effect of our gut microbiome on all of our health.